Right, so I want to welcome everyone back. This is going to be screencast number 2 for chapter 14. And what I want you to do is I want you to think about what we had looked at in our very first screencast for chapter 14, which was sort of giving you the basic introduction to Mendelian genetics. And so what we're going to do today is we're actually going to talk about various inheritance patterns that tend to be a little bit more complex than the simple dominant versus recessive inheritance that we had talked about in screencast number 1. So if you look at this screen, it says the relationship between genotype and phenotypes. Remember, genotype represents the genotypic arrangement for that particular characteristic, and phenotype is what you're going to see. It's rarely as simple as the garden peas that Mendel had studied back in the 1800s. Many heritable characters are not determined by only one gene. So in other words, there's typically either multiple genes, or we could have genes with more than actually 2 variations or alleles for that gene. So again, it can be a bit more complex. However, the principles of segregation and independent assortment that we had talked about in screencast number 1 even apply to the complex patterns of inheritance that we're going to talk about in today's screencast. So inheritance of characters by a single gene, again, may deviate from the simple Mendelian patterns in the following situations that you see down here. One of those would be considered a degree of dominance. In other words, there are some alleles that are not completely dominant or completely recessive. So the way that they're actually expressed in the organism is going to be based on degree. So when a gene has more than 2 alleles, again, this is something we had mentioned a bit earlier, obviously this can have a pretty significant impact on what you're going to see in the environment as well. Then we also have a gene that might produce actual multiple or many different phenotypes in that population as well. And so we're going to look at those in today's screencast. So the very first thing we're going to do is we're going to talk about the degrees of dominance. And so we actually have 3 different examples of this. So the very first one we're looking at here is something called complete dominance. And this is going to be represented when you have a heterozygote individual. So in other words, one that has both a capital and a lowercase letter, and a homozygote individual, again, having both the capital letters. The phenotype that you're going to see in those particular organisms are going to be identical. So in other words, that dominant allele is going to overshadow the recessive allele in the heterozygote condition. And then of course, we're only going to see the dominant characteristic in the homozygote condition because remember, because we have both of these alleles being the same. So what you're going to see in the population phenotypically is going to be exactly the same. The second example of dominance that we're going to look at today, which is a bit different from this first one, is something called incomplete dominance. And incomplete dominance basically means that the phenotype that you're going to notice in the F1 hybrid, or that F1 generation, is going to be somewhere in between the phenotypes of the 2 parental varieties. And so the easiest way to sort of illustrate this is to look over here on the right. They often use flowers, and so when you look at these 2 flowers at the top, we have one that's going to be red, one that's going to be white, and if you look at the red one, it has 2 capital letters, and we're using superscripts on these C's. The C is going to represent the color, and then of course the superscript is going to represent the variety of that gene. And so we have 2 capital R's, and in this one we have actually 2 W's. Sometimes when you talk about incomplete and codominance, they will actually sort of switch it up on you and not use capital and lowercase. They'll actually use 2 different letters. And so if you look down here, when the gametes are being produced by each individual, each one can only produce either C capital R or C W. So when you look at the F1 generation, you're going to notice it really doesn't look like the 2 parents, but it looks sort of in between. In other words, you don't have red, you don't have white, but you sort of have this pink color that's being represented. So it's sort of an incomplete variety between the 2 parents. And so that would represent incomplete dominance. And of course, when you allow this flower to self-pollinate, what you're going to notice is that instead of just getting 2 varieties, which is what you would normally see in complete dominance, what you're going to find is you actually have 3 varieties. And so the ratio is going to change a bit. So you have a possibility of getting a red flower. Here you can also get a pink flower, which is going to be that incomplete variety. Then you also, of course, have the possibility of getting a white flower. So that's going to be incomplete dominance. Codominance is a little bit different from incomplete dominance because this is represented when two dominant alleles affect the phenotype in separate but yet distinguishable ways. The dominant alleles are not necessarily more common in populations than the recessive alleles. And so what you see here, over here on the right, is a good example of codominance. And this is called a roan color. So this one that you see down here at the bottom is a roan colored horse. And it's a result of mating a white horse with a red horse. Now, what makes this one different is that if you look really closely at the hairs on this horse, the fur of this horse, what you're going to notice is that you actually have individual white hairs and you have individual red hairs. So they're both being represented in the same animal. That's different from incomplete dominance because this is considered an in-between. This does not have separate red pigment and white pigment making up this color. But yet down here at the bottom with this roan horse, you can see both dominant characteristics being represented in the same animal. So another good example of an alternative method of inheritance that you're going to find in the environment is something called multiple alleles. Most genes in the environment are actually going to exist probably in more than two allelic forms. Blood type is a really good example of multiple alleles. So you're going to notice over here on the right that when you look at blood type, we actually have three. And blood type is determined by a carbohydrate actually being attached to the red blood cell. And they represent those carbohydrates by letters. We have one that's represented by the letter A. We have a second represented by the letter B. And actually, the third allele actually does not have a letter. So it's actually missing that carbohydrate. If you have a genetic combination that actually has that capital A, then typically that person is going to have a blood type. If it has the B carbohydrate attached to the red blood cell, then it's going to be the B blood type. If you don't have either letter, then it's going to be considered what we would call an O blood type. Down here towards the bottom, these are the different combinations that you're going to find that's going to give you the different types of blood types that we find in the human population. The thing you're going to notice, though, is that when you look at the genotype, we actually have two different ways to get blood type A. You can have what we would typically call kind of like the homozygous condition where you have two capital A's, and that's going to give you blood type A. Or we have capital A and then, of course, no carbohydrate. So when you have these two genes come together, then it still represents a blood type A. Down here, you're going to notice the same thing for B. Two capital B's is going to give you blood type B. Or we have a B and then, of course, no carbohydrate. That's going to give you blood type B. Now, we do have a third and a fourth type of blood type in our population. We have one called AB blood type, and that's actually when you have both carbohydrates being attached to that red blood cell at the same time. And then if we have an individual that has neither A or B, then we classify that individual as blood type O. Now, another example of, again, an alternative to typical Mendelian inheritance is one called Pleiotropy. Now, most genes are going to have sort of multiple phenotypic effects on the individual. So remember when we talk about phenotypes, that's what we're going to see in the individual itself based on the genotype that that individual has. Now, we call that pleiotropy. Now, the easiest way to understand this is by looking at a certain hereditary disease or condition that you might find in the human population. Cystic fibrosis and sickle cell disease are actually good examples of this. So what you want to think about here is when you're looking at, for example, an individual that has the sickle cell gene, basically the single gene on their chromosome, if they have sickle cell disease, instead of only affecting maybe one characteristic for that individual, sometimes you're going to notice that in a pleiotropy type of situation, especially again for diseases, it actually affects multiple parts of the body. So in this case, if you have sickle cell anemia because you have this condition, there's a good chance it might affect your eyesight, so there's a possibility you could go blind. You could possibly have some situations that could affect your liver, so you could possibly go into liver failure. And then, of course, we have situations with sickle cell persons that actually have problems with their heart as well, and so they could possibly have a heart attack. So this is an example of pleiotropy. Now, another thing we want to make sure we talk about is something called epistasis. And sometimes when we talk about Mendelian genetics, we're going to look at actually two or more genes and sort of how they interact with each other. So when you talk about epistasis, this is going to be a gene at one locus or one position on that chromosome that's going to alter the phenotypic expression of a gene at a second locus on a different chromosome. So in other words, we have two genes kind of influencing each other. So a good example of this would be in mice and actually lots of other mammals. We're going to look at coat color, in other words, the color of their fur. This actually depends on two genes. In other words, one gene is going to determine the actual pigment color. And so if we look at this example, we could look at the capital B representing black and the lowercase b representing brown. But there's going to be a second gene, and this gene is going to actually code for whether or not the animal itself actually has color, or C or lowercase c, for no color. So this is going to determine actually whether or not the pigment itself is going to be deposited in the hair. So one's going to determine the color, and then one's going to determine the actual deposition of the pigment. So it's going to show up in the phenotype. Now, when you look at these two genes, it's like the typical two-trait cross that we had looked at in the first screencast, but this just sort of gives you an idea of how they're influencing each other and what you're seeing. So again, if you have that capital B, then you're going to get sort of a dark black color mouse. If you have two lowercase b's, then you're going to end up getting the brown mouse. But if you notice, again, if we don't have any deposition of color, then you're going to end up getting a mouse that actually does not have any pigment in its fur. So we actually end up with not just two phenotypes, but we have a third phenotype, and that's all based on the influence of that deposition gene for that fur color. Another thing that we need to mention is something called polygenic inheritance. We're not talking about pleiotropy, so don't get those two words confused. Polygenic, we have a prefix here that means many, so we have many genes actually influencing the characteristic that we see. So quantitative characters are those that vary in the population along what we consider a continuum. Now, a continuum is basically a range. So quantitative variation usually indicates some form of polygenic inheritance. So in other words, the additive effect of two or more genes on a single phenotype. So skin color is a really good example in humans of a polygenic inheritance. And so if you notice, if you look all the way up here in the left-hand corner, this would represent an individual that actually has a very light skin color. And then if you notice down here at the very bottom right-hand corner, this would represent an individual that has a very dark skin color. And if you notice, there is a continuum of skin color, many different types of skin color that you see in this Punnett square. And so when you talk about polygenic inheritance, basically it means the number of dark skin alleles that you have. If you have more, then you're going to end up having a darker skin. And then if you have either none or few dark skin alleles, then that's going to represent an individual that has a lighter skin color. So this is considered polygenic inheritance. It's all based on the number of dark skin or number of a certain type of allele that you have in that genotype. That's going to determine the phenotype of that individual. Now, something that's really important for us not to forget about is that the environment can have a huge impact on the phenotype that we actually see in an organism. So we need to consider Mendelian genetics, and again, it's really important when it comes down to the phenotypes that we're going to see, but sometimes the environment can really have a significant impact. So the norm of reaction is the phenotypic range of a genotype as it's influenced by the environment itself. Now, the hydrangea flower is actually a good example of this, because even though we have genetically determined colors in these particular flowers, based on the acidity of the soil, that can influence whether or not this flower ends up being a really deep pink color or actually a very purple or sort of a blue color. And it's all based on whether or not the soil is either very acidic or very basic. So norms of reaction are generally broadest for polygenic characters. So such characters are called multifactorial because genetic and, again, environmental factors are going to collectively influence the phenotype that you're going to see in that particular organism. So there's actually lots of human traits that follow the Mendelian patterns of inheritance that we've looked at. The unfortunate thing is that humans tend not to be really good subjects for genetic research because first, our generation time is extremely long. In other words, we don't live for just a couple of years or a couple of months. We actually live for between usually 65, 70, 80, 90 years. And so that generation time is really, really too long for really quantitative study. The parents produce relatively few offspring. So what you get from your subjects can give us a little bit of information, but you need a larger sample size to really make some definitive conclusions on what you're looking at. And of course, breeding experiments would be totally unacceptable when it comes down to humans. However, basic Mendelian genetics endures as the foundation of human genetics because we're like any other organism on this planet. We do follow those patterns of inheritance. So what we do is we actually use something called a pedigree. And a pedigree is simply sort of like a family tree that's going to describe the interrelationships of parents and children across the generations. So what we do is we kind of go back in the past, we look at somebody's family, we look at the characteristics that they have, and we can create this pedigree. And once we create the pedigree, we can sort of use the pedigree to kind of help us predict what might happen in the future. So inheritance patterns of particular traits can be traced, like we had said, and they can be described using those pedigrees. So this would be a good example of a pedigree, and these are things that you will work with in class. The first thing you want to notice is that first we always identify whether or not the individual is male or female. When you see a shape, either the square or the circle, that is filled in, that's going to determine whether or not it's affected or unaffected. So if it's filled in, it's considered affective. So if you look over here, you're going to notice this line is going to connect these two individuals. That's going to basically indicate to us that would be a mating between those two individuals. And then, of course, these vertical lines that you see, which would eventually branch from these parents, are going to be the offspring, and they typically follow a birth order, with the firstborn being on the left. So this would be considered our first generation in this pedigree. These would be the grandparents, and so you can see we have the genotypes represented here. The second generation, of course, would be the children that are produced from this first generation. So these would be the parents, the aunts, the uncles. Again, genotypes are being represented. And then we would have the third generation, which would be represented by these two sisters. So these are two different families. And if you notice, they came together right here. And if you notice, we have two affected individuals right here. And in this case, we're looking at the widow's peak. In other words, that sort of kind of vampire-looking hairline that you might see in some individuals. And so in this case, because it has a capital W, remember this is just going to be typical dominant recessive inheritance, these individuals actually do have that characteristic. So if you notice down here, for these two sisters, we have one individual actually that has the widow's peak and one that does not. Now, for some of us, we'd look at that and wonder, well, why in the world did that happen? Because both of these individuals actually have a widow's peak. Well, you got to remember, if that individual is heterozygous, then there's a possibility we would have two lowercase w's actually come together. So down here towards the bottom would be another example of a pedigree. So again, this is going to represent our first generation. So these would be the grandparents. And again, you notice we do have the genotype of what we're looking at right below each of the shapes. Second generation would be the parents, the aunts, and the uncles, just the same as we had seen up above. And in this case, we're looking at whether somebody has attached or free earlobes. And so if you notice, for this third generation, again, for these two sisters, for somebody who is affected, it's been determined that this would be a recessive characteristic. So somebody who has attached earlobes, in other words, they don't hang free, they actually have two lowercase letters. Now in this case, we actually had this produced from two individuals that had free earlobes. So if you notice, these two came together because each of these individuals right here were heterozygous for the condition. So that would mean this person on the right would either be two capital F's or a lowercase f and an uppercase f. And the thing about it is we're not quite sure because both of these individuals actually were heterozygous for the condition. So we can't give a definitive conclusion whether or not this individual is one or the other. So what we're going to do next is we're just going to give a really brief overview of a couple of recessively inherited disorders and a couple of dominantly recessive disorders. Recessive inherited disorders tend to show up in individuals that are homozygous for the allele. Again, that would be two lowercase letters. Those individuals out there that actually have one of those recessive alleles, in other words, they'd be considered heterozygous, oftentimes we will use the word carrier. In other words, you're not seeing the expression, of course, of that recessive characteristic, but we call them carriers because they could potentially pass it on to their offspring. Albinism is a good example of a recessively inherited disorder. So albinism is simply the lack of pigmentation in the skin and the hair, and you can see this over here on the right side. If a recessive allele that causes a disease is rare, then the chance of two carriers actually meeting and mating is typically pretty low. Matings between close relatives do tend to increase the chance of mating between two carriers of the same rare alleles, and simply because you have more of an opportunity if that recessive allele is already in the family of actually bringing those two individuals together. Over here again on the right you can see albinism, and again since it's a recessive disorder, the only way we're actually going to get somebody who has this condition is if we have two recessive alleles actually come together. Cystic fibrosis, again, is another good example of a recessively inherited disorder. It's actually the most common lethal genetic disease in the United States. It strikes 1 out of every 2,500 people of European descent. It results in a defective or absent chloride transport channel in the plasma membranes, typically in the respiratory system. Some of the symptoms that you would find with people with cystic fibrosis would be mucus buildup, and especially the respiratory system or the lungs of the individual. But that mucus buildup can also occur in other internal organs. And they also have an abnormal absorption of nutrients in the small intestine. Sickle cell disease or sickle cell anemia is one we've already talked about. Again, it affects 1 out of every 400. This disease is caused by the substitution of a single amino acid in the hemoglobin protein of red blood cells. And again, as we had said, it tends to cause physical weakness, pain, again, lots of different phenotypic expressions of this disease, organ damage, and then of course it could even cause paralysis depending on where it's affecting the individual in the body. So the flip side of this is dominantly inherited disorders. Some human disorders, believe it or not, are actually caused by dominant alleles. Dominant alleles that cause a lethal disease are pretty rare, and typically what they do is they tend to arise or come about simply by mutations. Achondroplasia, this is a form of dwarfism, is caused by a very rare dominant allele. So if you look over here on the right, this Punnett square is representing what you might expect if you have one parent who is a dwarf for this particular type of dwarfism and one parent that is normal. If you notice, they have achondroplasia because they have at least one dominant allele, because it's a dominantly inherited disorder. You don't have to have two, you actually only need one for it to be expressed. So this person is producing children with a normally heighted individual, and in this case, if you're normally heighted, you have two recessive alleles, and if you notice in this case for their children, what they could expect is there is a 50-50 chance that their child produced would actually have this form of dwarfism. Now, Huntington's disease is another good example of a dominantly inherited disorder. This is a degenerative disease of the nervous system. The problem that we have with this disease is it's a bit more common than others, simply because oftentimes we tend to pass this particular dominant allele to our offspring, simply because the effects of Huntington's typically don't show up till around 35 to 40 years of age, and it is considered a lethal type of disorder, so people die from this. And the thing here is that by the time you're 35 or 40, for most people, if you're going to have children, typically you've done this by now. And if you have no obvious phenotypic effects up to this point, you really don't know if you really don't realize that you could be passing this on to your children. And so this is where, if you know that you do have a family history of Huntington's disease, it's really good to have a genetic panel done to see if you actually carry this dominant allele. Because if you do, then that would be something that definitely you would not want to pass to your offspring. All right, so that's going to finish up this second and final screencast for Chapter 14. As always, please make sure that you have completed your screencast study guide.